So the first one, we're going to have excellent speaker, Selene Lomoio. So she will discuss the protein known as Gga3 and how this protein contribute to AD pathogenesis through the control of base-1 trafficking. Selene actually crossed the Atlantic Ocean to Tufts University, joined Josephina's lab for postdoctoral training. And she has done a great job and promoted to research assistant professor very recently. And today she's going to speak some of the study just published recently in Sci Translational Medicine. So welcome, Selene.
Thank you so much. Thank you for the nice presentation. I'm gonna share my screen. And I'm gonna just do another couple of things. Can you see my screen?
Yes.
Thank you so much. Thank you again to the Open Box Science Organization and Jerry for organizing this nice seminar. Thank you, Dr. Yan, for accepting our invitation of being a moderator here. But I want to also thank you, Leah, for being, here to share this space with me today. The story I want to tell you today regards a paper that we recently published in Science Translational Medicine this past November, and is the story of how Gga3 regulates base axonal trafficking. In order to do that, I like to introduce a couple of concepts or revise them for people that are already familiar with them. Alzheimer's disease is the most common form of dementia, and when you look at the brain on Alzheimer's disease patients, you can recognize histopathological hallmarks of the pathology. Among them, you have beta amyloid plaques, neurofibrillary tangles, and dystrophic neurites. Today, I like to focus my attention on dystrophic neurites. Dystrophic neurites are these axonal defects that may reflect transport problem and can be divided into three different categories. You have those neurites, dystrophic neurites, that are associated with amyloid within senile plaques. You have some that are associated with neurofibrillary tangles. But then you have a third category where these axonal defects are specially distinct from the hallmarks lesion of AD. When you look at this third category, they will look like this. This is a human AD brain, Braak stage 2. And in this area, as you can notice, there is no extracellular A-beta deposition, and you have axons bearing these enlargements. These enlargements are focal axonal swelling that correspond to abnormal accumulation of axonal cargos and transport proteins. It's very important to remember that these kind of abnormalities develop and form very early during the progression of Alzheimer's disease. It's also important to add one more detail to this story. We now know, thanks to many studies and scientists, that increased plasma concentration of neurofibrillary light chain is indicative of axonal damage and has been detected in AD presymptomatic stages. All of this is telling us that when we look at axonal dystrophy, we look at the hallmark of the pathology that is it's happening and is starting very, very early during the pathophysiology of Alzheimer, and can also be detected and spotted by using neurofilament light chain as a marker of axonal damage. It's also important to remember that many proteins that are associated with the pathogenesis of Alzheimer's disease have been observed in the axonal compartment of neurons. Many of them have been found at the presynaptic terminals. And of course, in order for them to be delivered to the final destination and the axon, transport is a key player in this mechanism. Why this is important? This is important because among these proteins, you have the beta secretase. The beta secretase, also known as BASE1, is the rate-limiting enzyme in the generation of Alzheimer's disease. In normal condition, BASE1 localizes to presynaptic terminals and accumulates instead in dystrophic neurites around amyloid plaques in Alzheimer's disease brains. No changes in the levels of the messenger RNA have been associated with this elevation. So we scientists strongly believe that this elevation is most likely due to post-translational modification. In order to proceed with the story, I need to introduce another player. This player is Gga3. Gga3 is a monomeric clathrin adaptor protein highly expressed in the brain, and has been shown to bind base and drive it to the lysosome for degradation. Through the years, our labs accumulated evidences about base and Gga3. We now know that Gga3 levels are decreased and inversely correlated to base 1 in human AD temporal cortex. We do know that base levels are increased in the brain of Gga3 non-mice. And recently, my colleague Won-Hee Kim described that the lack of Gga3 exacerbates beta-amyloid pathology in the brain of a mouse model of familial Alzheimer's disease. From this data, it's obvious that we knew already that Gga3 was expressed in the brain, but exactly where in the neurons, we had no idea. We set out to find out where Gga3 localized in the neuronal compartments, and from the images where we overexpress the GFP Gga3, it's immediately evident that, sorry about this, that Gga3 localizes both through the dendrites and axons. In order to verify that even the endogenous protein was distributed in this way, we polarize our neurons in microfluidics devices, obtain and lysate the cell side and the axonal side, and we run a Western blot of these two separate fractions. And this allowed us to demonstrate for the first time that even Gga3 at the endogenous level, so the endogenous protein, is in fact present in the axonal compartment of neurons. At this point, we knew for sure that both base and Gga3 were in the axon, but we didn't know anything about their spatial relationship. We did overexpress both proteins together in axons, calculated a Mander coefficient of localization, and found out that the two proteins are, in fact, very close, both in dendrites and axons. Being very close, though, doesn't mean that, they are co-transported. So in order to assess if these two proteins are actually in the same vesicle and they move together along the axon, we went for live cell imaging experiments. In this movie, you have on top a GFP Gga3 axon. At the bottom, you have a base and cherry positive axon. And it's immediately obvious that both proteins are capable of bidirectional movements along the axon. Gga3 is a slower protein compared to base, and while base is evenly distributed among anterograde and retrograde movement, Gga3 is mainly retrograde directed. From the overlay, it was obvious that there were many vesicles that were carrying both base and Gga3. And when we quantified this vesicle, we found out that 37% of base 1 vesicle are in fact co-transported with Gga3. It's important to note that the majority of this vesicle was retrograde directed. No surprise to us, because if you consider that the role of Gga3 is traffic base back to the soma, where it's normally degraded for degradation in the lysosome, this makes sense that the majority of base and Gga3 is co-transported in a retrograde manner. So from the axon back to the soma. As I mentioned before, we knew that when you delete Gga3 in a mouse brain, base 1 gets elevated. We didn't know, again, exactly where this elevation was taking place. And in this image, you have two neurons, a wild type, Gga3 wild type neurons, and a knockout neurons, where we overexpress base 1. And from this overexpression, it was immediately obvious that base 1 was elevated in the knockout neurons, specifically at the axonal level. In order to verify that this was true even for the endogenous base, we took advantage of the transgenic mouse. The mouse, we have both wild type and knockout mice in our lab. And the area we focus on are the mossy fibers for two reasons. Mossy fibers are a bundle of axons, and the base 1 is enriched in this area. And this is, as is evident from this image and confirmed by the quantification, even at the endogenous level, base 1 is elevated in the axons of Gga3 knockout neurons. Since we knew at this point that 40% of base vesicles are co-transported with Gga3, we ask the question, what's going to happen to the trafficking of base in the axon when Gga3 is not present? And here are two movies. On top, you have a wild-type neuron with base moving along the axon. At the bottom, same condition, same experimental setup on a knockout neuron. What is evident is that in the knockout neurons, base vesicle cannot move at all along the axon, and it's confirmed by these chymographs and analysis. The thing that surprised us was that both the anterograde and retrograde trafficking were affected. I like for you to remember this consideration that for us was a surprise that even the anterograde trafficking was affected, because we were expecting only the retrograde trafficking to be disrupted by the lack of Gga3, because this will be useful in a couple of slides. Looking closer at this axon expressing base and cherry, where base and cherry was basically not moving. If you look closer then, you will see that base and cherry is stuck there and start accumulating in this swollen area along the axon that clearly resemble the dystrophic neurites that have been found in many neurodegenerative pathology and Alzheimer's disease. In this specific movie, I was very lucky and I was able to record live the formation of a new accumulation of base 1 in the axon. And then if you look inside the swelling instead, you notice that the base vesicles are literally trapped there. They can only bounce inside the swelling, but they cannot leave the swelling. Again, we took advantage of the mice, went back to the brain of these mice, wild type and knockout, to check for axonopathy. The first area we considered is actually the cerebellum. You would say that the cerebellum is not very typical area to consider for Alzheimer's disease, but is a perfect area to study axonal pathology. The cerebellum is very well organized, especially if you stay for Calvindin. It will give you this perfect depict image of Purkinje cells. Purkinje cells have a soma here in single layer, dendritic arborization, and axons in the internal granular layer. When we look at the axon, we immediately notice that in Gga3 knockout cerebella, there was this accumulation of enlargements. And when we quantified it, we found out that the swelling density was increased by 3-fold in the cerebella of knockout mice compared to control littermates. We then switched to an area that is more common in Alzheimer's disease field, and this area is the hippocampus. We did use Bielskowski silver staining, so modified silver staining, to depict and stain axons. Axons in this area of the hippocampus, the CA1 area, specifically the stratum lacunosum moleculare, are really, really well organized. They're tightly packed in these little islands, and the architecture is very typical and characteristic. When you are in a knockout condition, what you immediately notice is a total loss of this perfect organization. And when you quantify the number of axons, you will also, we also realize that there was a reduction in axonal density in the knockout mice compared to the controls. When I said to you a couple of slides ago that we were surprised to see that both the anterograde and retrograde trafficking were disrupted, it's because our hypothesis was, since base is co-transported with Gga3 and is mostly in this situation retrograde directed, because the role of Gga3 transporting base 1 to back to the soma for degradation, why is the anterograde trafficking affected if Gga3 is not there? Our hypothesis was, when base accumulates in the axon, has an higher chance of cleaving the substrates. Among these substrates, you have APP. And the higher cleavage of APP will induce an accumulation of A beta oligomers, most likely intracellularly at this point, that will further disrupt the axonal trafficking. In order to verify our hypothesis, we decided to block the generation of A beta oligomers by using beta and gamma secretase inhibitors in the form of C3 and DAPT. And look out for those two phenotypes that are typical and have been described for base in Gga3 knockout neurons. And in fact, when you block the production of A beta oligomers by using this inhibition, both the swelling phenotypes and the trafficking dysfunction can actually be prevented. Together with our collaborator Dan and Merck, and Matt Kennedy was the person who coordinated all of this, we decided to treat live animals with a potent compound, and Gga3 is a BACE inhibitor widely characterized by Merck, by the Merck colleagues. And we started to treat mice at 2 months of age for 8 weeks. After these 8 weeks, we analyzed cerebellar swelling density and CA1 axonal density. And we asked the question, can BACE inhibition prevent this axonopathy that we observed before in Gga3 knockout mice? And from this quantification, it's obvious that yes, BACE inhibition early in the pathology at 2 months of age can in fact prevent the formation of cerebellar axonal density, starting with the lowest dose of the treatment, the 3 mpk, up to the 30, while in the hippocampus, only the highest dose was able to prevent axonal degeneration and reduction of axonal density. Finally, I just mentioned before that my colleague Won-Hee Kim was the one demonstrated that in 5X Gga3 knockout mice, you have an exacerbation of A beta pathology. And thanks to this, to his inputs, I asked myself, what is going to happen to an axonopathy, an axonal pathology in the brain of 5X FAD mice if I delete Gga3? And so we did use our Gga3, 5X, Gga3 wild type and knockout mice at 2 months of age to investigate this question. At 2 months of age, in 5X Gga3 mice, you almost have no extracellular deposition, especially in the area of the cortex where we ran these studies. We immediately noticed that when you delete Gga3, and so you have a 5X Gga3 knockout mouse, the number of swellings that are independent from A beta extracellular deposition are increased in these mice compared to control litter mice. When we looked closer, we also found out that in these swelling that are here highlighted in this inset and stain for neurofilament, you have an accumulation of endogenous base at 2 months of age when you delete Gga3. So for the second time through this year, we demonstrated that the deletion of Gga3 has an effect in accelerating a pathology that is related to AD. But in this case, this pathology is pre-plaque, is happening before the pre-plaque phase of the neurodegeneration. And finally, together with our collaborators, Rudy Tanzi and Dmitry Prokopenko, I hope I'm saying both last names correctly, we identified a novel AD-linked Gga3 variant. This variant is an in-frame indel resulting in the insertion of a threonine at position 545 of the long isoform of Gga3 and was present in 6 out of 8 affected individuals from 4 families with late onset Alzheimer's disease. All families are of African American ethnicity and they belong to the African population. And so this indicates that this rare variant appears to strongly concentrate and co-segregate with late onset Alzheimer's disease in the African American population and patients. Once we identified these variants, we decided to check if these variants was able to rescue the phenotypes that we observe in Gga3 knockout neurons as the wild type Gga3 does. And actually by overexpressing, reintroducing insertion 545 in the knockout condition, we weren't able to rescue the swelling phenotype or to maintain a proper trafficking of base. Only the reintroduction of the Gga3 wild type or the reintroduction of the insertion 545 and a base inhibitor was able to rescue the status of base 1 trafficking and swelling back to normal. And with this, I like to summarize what the mechanism we are proposing is. We propose that Gga3 loss of function induces an impairment of base 1 retrograde trafficking by affecting the subset of vesicles that co-transport both proteins and are mainly retrograde directed. As a consequence, base 1 cannot be trafficked back to the soma where it's normally degraded and starts accumulating in the axon. Accumulation of base 1, as I mentioned, would be predictive of enhanced amylodogenic cleavage of APP and increased A beta accumulation. And this finally induce base dysfunctional terograde transport that is most likely secondary to increased axonal production of A beta. And in turn, this will lead to the formation of axon as well. We strongly believe, just to conclude, that our study and our data support the hypothesis that loss of function of Gga3 can be an initiating event in AD pathophysiology. We also have data that that give a possible explanation for the prevalence of axonopathy during AD early stages, in which axonal base elevation is not a consequence of A beta extracellular accumulation and plaque formation, but is an earlier and upstream event that occurs during the pre-plaque phase of the pathology. And finally, the in vivo base inhibition suggests that base inhibitors can prevent axonal damage in the absence of extracellular deposition. At least that's what our data suggest, if used early enough in the pathology. And with this, I like to thank really all the people who made this work possible. It was a monumental work, and Giuseppina, our mentor, when he came, Rachel Willen, Kevin O, and our Robinson that contributed to this paper greatly. And then our collaborators, Rudy Tansi, Dmitry Prokopenko, and Matt Kennedy. We were lucky enough to have fellow scientists sharing their reagents with us. And a special thanks go to Liding, who supported me through the live imaging macros generation and live cell imaging analysis, and of course, our funding. And with this, I'm done and up for questions.
Thank you, Selene. Now we open for the panel for question. I mean, anyone has question, you can either type it or you can, I don't know whether we can unmute them to ask a question.
I see there is a question in the chat from Jerry.
Oh, you can see it? Okay, that's good.
If you want, I can address this question. Jerry is asking if we have looked at astrocytic and microglia response in Gga3 knockout 5x FAD mice. And this is really a good question, but no, not for this round of experiments. So I actually have no data regarding astrocytic and microglia responses at 2 months in these conditions. But yes, it's a question that should be addressed. I agree with you. I really agree with you.
So Selene, my lab is very interested in base 1 trafficking as well. So we worked on a protein reticulin 3, which regulate the base axonal trafficking as well. Mm-hmm. Do you know, I mean, Gga3 and the reticulin 3 have any coordinated effect or not?
Not so. Honestly, I don't know if they have a coordinating effect in regulating the trafficking of base. I know very well your studies on reticulin 3, and I agree that there must be more than one protein coordinating base 1 trafficking. I'm sure of that. But I'm not aware of any direct connection between Gga3 and reticulin 3 in coordinating the trafficking of base so far. But I don't exclude they may have an interaction in managing this trafficking.
So another question related to the question is, you saw base 1 accumulated in the axon.
Yeah.
So the question I have is, how do you know the base accumulated there actually is functional?
Yes, you are absolutely right. So our data suggests, so the data that we have after treatment with base inhibitors, and the fact that when you inhibit base, you have an improvement actually of the trafficking of base itself, suggests that in fact the base that is accumulating in the axon is functional. we believe that base that is accumulating in the axon is functional because the data on the because of the data with the beta secretase inhibitors, but we didn't run any real functional assay. So if I don't have direct evidence for that.
Okay, thank you. There's another question in the chat box.
Oh yes, I have a question that say, are you using global knockout in your study? Do other brain cells express Gga3? Yes, our Gga3 knockout mice are global knockout, they're not conditional knockout, they're not any of that. But regarding the question about brain cell express Gga3, all of them, honestly, I'm sure that neurons express Gga3. I don't know about astrocytes at this point. In my hands, in my mind, I cannot give you an answer regarding other brain cells. At this point, we do know though that neurons express Gga3. Keep in mind that there are other two isoforms of the GGAs, and another one is highly enriched in the brain, and this GJ1. So it may be, I'm not sure, it's just an hypothesis, I'm just speculating here. It could be that GJ1 and GJ3 are expressed, in fact, in different cell types. And then I have another question that is asking, am I wondering if you have tested if the GJ3 knockout mice have some cerebellum dependent phenotypes, such as ataxia, as the knockout clearly causes axonal pathology in the cerebellum, perhaps at the granular neuronal axons? Yes, I do agree with you. Thank you for this question. I wonder myself if these mice display any fine coordination issue, or as far as I know, we extensively, I didn't, but a colleague of mine did a extensive battery of behavioral tests on these mice, and apparently the main motor skills of these mice are fine. But also keep in mind that, as you know, the cerebellum is the plasticity in the cerebellum is incredible. So those degeneration that we're seeing at the Purkinje cell level may not cause any evident symptoms at the cerebellar and motor coordination level. It may take probably many more months if this pathology progresses in order to reach an evident defect that is cerebellar driven in this kind of mice. And then I have another question. Does this variant identified in African American show reduced expression of Gga3, both messenger RNA and protein, or it is related to post-translational modification of Gga3? So I'm not sure I got the question correct, in the correct way. So what I can tell you is when we don't know. So this variant, we don't know if it's how it's causing this loss of function, okay? So far, we don't have enough data to demonstrate, to speculate about how this variant is inducing the loss of function. I don't have data regarding the messenger RNA and protein level, but it could be due actually to post-translational modifications, as you were mentioning in your question. I hope these answer your questions here.